Hi students, myself Dipankar Majumdar. I am Assistant Professor of Department of Microbiology. Today I will discuss regarding the light microscopy that is included in the second semester of NEP 2020. Okay. We, the human beings, are able to see the objects around us, in us, on us, with the help of our eyes. So many objects that are very tiny are not able to be seen with our eyes. So, to see the tiny objects like bacteria, viruses, fungus, or the cells, we have to use some specialized instruments. And this is simply known as the microscope. And today my lecture is on light microscope. That means microscope where the light, especially the visible light that is used to magnify the tiny objects that can be seen with the help of microscope. Okay? Now, the word microscope is derived from two Greek words. Micro, that means a small, plus scopia, that is to see. Okay. So, the small objects or small living organisms that are seen with the instrument is known as microscope. Ancient Greek people were aware about the use of glass, and they used glass that is filled with water to magnify the objects. That is the water-filled glass globes, and the curvature of that globe is used to magnify the objects. And this type of setup was used to see the insects, and in Greek, Phili. Phili means insect. So it was simply known as the Phili microscope. And another word that is very much used, that is lens in microscope. And this term lens is derived from Greek word lentil. Lentil is a bean, and its shape is like that. So the word lens is derived from the lentil bean from Greek. Now, the compound light microscope, that means in which the microscope where both objective lens and ocular lens is used. That means more than one lens is used, so it is simply known as compound microscope. These are the various types of microscopes, from primitive, that is from Leeuwenhoek's microscope, and Winkel-Zeiss microscope, British microscope, etc. and the present days Zeiss Primostar microscope. Okay, these are the various types of light microscopes. Another history. First microscope was manufactured by Hans and Zacharias Janssen. In 1590, they were the Dutch eyeglass makers. First compound microscope that was made by them. Then Robert Hooke, he is famous for his microscope, and he introduced the term cell from the word cella. And then Anton van Leeuwenhoek was the Dutch spectacle maker, and he is simply known as the father of microbiology. These are the timeline of the development of various types of microscopes. Yeah, these are the types of microscopes. Microscopes are divided into 3 categories: light microscope, scanning probe, this is a special type of microscope, and electron microscope. And in case of light microscopes, bright field, dark field, fluorescence, phase contrast, confocal, stereo, video, polarizing, UV interference, all these are under the light microscope, where the visible range of light is used as the source of illumination, except in UV, where UV light is used. And special type of microscope, that is scanning probe, atomic force microscope, scanning tunneling microscope, scanning thermal microscope, scanning ion conductor microscope, and last one is electron microscope. These are basically 3 types: transmission electron microscope, scanning electron microscope, and scanning transmission electron microscope. Okay, the light. This is the light, and this is the wavelength of light. Here is elevation, crest, trough, amplitude, all these things. Yeah. Light rays is simply known as electromagnetic wave. That means it is having 2 components, that is electric field and magnetic component. So light wave is known as electromagnetic wave. And this is another diagram. This is more detailed diagram. Yeah. This is the dispersion of white light, that is visible light, through glass prism. And this, after dispersion, the light is separated into 7 different colored lights, that is simply known as Vibgyor, and each type of light is used in microscope, and they are having different wavelengths. And the range of the wavelength of visible light is from 400 nanometer to 780 nanometer. Yeah. This is the type of electromagnetic radiation. Here is the UV rays, gamma rays, X-rays, infrared rays, radio waves. From left side to right side, the wavelength of light is increasing. But from right to left, the ionization energy is increasing. This is the electromagnetic spectrum, and these are simplified to magnify various types of objects, that is in radio wave, buildings, human beings, honeybee, pinpoint, protozoans, molecules, atoms, atomic nuclei. All these are shown here. Okay. From right to left, short wavelength, high energy, high frequency. In the right direction, long wavelength, long energy, and low frequency. Yeah, this is a Zeiss. This company is the one of the best microscopic manufacturing company of the world. And Carl Zeiss was an instrument manufacturer in the city of Jena in Germany. And their caption is, "We make it visible." Yeah. This is for compound light microscopy. The basic question is, when should we use microscope? To see a mango tree, or to see a building, or a human beings, or small organisms like ants, we don't need microscope. Because our naked eye is able to see or able to resolve these objects. So our eye is having some limitations. What is the limitation? This is here. This is simply known as the limit of resolution. Okay. Here, 2 points are shown here, P1 and P2. The power of the microscope depends on which is that when 2 separated points can be resolved as 2 points. Here it is 2 points. This is much more closer, but we are able to distinguish them as 2 separate points here. The points becomes more closer. Here also more closer, but we are able to distinct these 2 points as 2 separate points. But here the 2 points are overlapped. So that means our naked eye is not able to see the 2 points as 2 separate points, or our naked eye is not able to distinguish between the 2 separate points as 2 separate points. In that case, we have to use microscope. Okay. This is another diagram. This is the resolution. These are the overlapped points. This is less overlapped, and these are the 2 points. Those are separate from each other. This is the equation upon which the microscopic resolution can be calculated. D is the limit of resolution. Lambda is the wavelength of light used. Na means numerical aperture, and that is calculated by n sin theta. N means the medium that is used. Okay, this is objective lens. And here the light rays that is passing through the specimen slide, and it creates an angle. Theta is the half angle. Okay, so and the medium that is used is air. Here air is present, and its refractive index is 1. Okay, and that means Na is equal to n sin theta. Okay, n is 1, and the maximum value of sin theta is sin 90 degree. So here Na is 1. But in practical, we will not be able to create the value of theta that is 90 degree. In theory, it is possible, but in practical, it is not possible. You can try it. You will not be able to do this. Another thing is, objective lens. Okay, I am coming to that point. Yeah, this is the various limits or sizes of the living organisms. Okay, from ant to small molecules. Abbey's resolution limit or diffraction limit is 0.2 micron. Okay, these are the parts of the microscope and their function. Yeah, various parts are present, and the parts are divided into 2 major parts. One is mechanical parts, another one is optical parts. In optical parts, condenser is present, objective lens is present, ocular lens is present. Okay. And these are the various types of lenses present. This is the numerical aperture of the microscope what I am discussing. Yeah. Here I have discussed, and here the objective lens, that is magnification with 100x. That is simply known as the oil immersion lens. Okay, oil immersion lens. Why we are using oil immersion? Various types of oils or solutions are present in the market, because in slide glass is used. It is having refractive index mu is equal to 1.51. Here objective lens. Lens is made up also of glass. So it is also having the refractive index of 1.51. And here there is a gap. This gap is filled with air. When the medium is changed, or the refractive index of the two different medium is different, at that time light may diffract. That means that is bending of light, and this may cause the loss of light. So we have to minimize. We have to minimize this. In case of 100x objectives, its length is much more higher, and the lens is made up of elements. Elements means the small lenses that are fused. So the lens that is having higher magnification will have higher number of object elements. In case of 4x, the number of elements are lesser. In case of 10x, it is quite higher. 40x, it is higher. And in 100x, many lenses are fused to form 100x objective lens, and that's length is much more higher. Okay. So the objective lens of 100x magnification is near about to the specimen slide, and the gap is minimum. So you are able to fill the gap with a type of oil that is simply known as immersion oil. So in case of 100x, oil immersion can be used. So the lens of 100x magnification is simply known as oil immersion lens. And oil immersion is having the refractive index. Oil immersion mu is equal to 1.51. That means it is filled with the oil that is having the equal refractive index with that glass and that specimen glass. So there will be no bending of light. Light will directly enter into the objective lens. So the magnification will be much more brighter, much more clear. For that reason, oil immersion is used here. And for compound light microscope, in case of 4x, 10x, 40x, you will not be able to use the oil immersion because the gap is much more higher. But in case of, so these 4x, 10x and 40x lenses are simply known as dry objectives. 4x, 10x and 40x are known as dry objectives, and 100x is known as oil immersion objectives. And another thing is, in case of microscopic mirror, when you are not using auto illumination microscope, in case of simple compound light microscope, the mirror is plano-concave in nature. That means one side is plane, another side is concave. Why this type of mirror is used? Why not it is both side plane, biconcave, or another type of lens? This is having some role in microscope. When you are using sunlight, source of illumination, at that time, the gathering of light is occurred, and then after gathering, a beam of light is formed that is passing through the condenser, then through the specimen, and then entering into the objective lens, and then magnification is occurred, and after that ocular lens magnifies the final image. Okay. And in case of plain sight, it is used when you are using artificial light or light bulb or lamp, whatever may be. In that case, the source of light is nearer. So there is no need to gather the light, only the reflection of the light is needed. At that time, the plain sight is used. Yeah, this is for the use of oil immersion. Now I am coming to the phase contrast microscope. Yeah. In case of light microscope, there are some limitations. In light microscope, the fixation is needed. Fixation may be of heat fixing or chemical fixing that kills microbes. And stain is applied. And stain is a hazardous chemical that may kill the microbes and alters the shape of the microorganisms or cells. To overcome this problem and to see the unstained and live cells, scientists tried, and phase contrast microscope was invented. And this microscope is having some advantage that the microscope can create image of the live specimen and unstained specimen. So you will be able to see the specimen in its exact shape as well as in its exact activity. The Dutch scientist Fritz Zarnik Dutch physicist. That is, he is from the Netherlands. Okay. In 1953, he was awarded Nobel Prize for the invention of phase contrast optics, NP 1953 in physics, for phase contrast optics. Yeah. We are able to see the object with the help of light. Same wavelength light is having different type of brightness, and this brightness or dullness can be perceived by our naked eyes, and this is due to the difference in amplitude. Okay. In phase contrast microscope, what it is done, the biological material is able to retard the wavelength of light into lambda by 4. Lambda by 4. Suppose the wavelength of light is lambda. That may be retarded by biological sample, that is lambda by 4. And this change is not perceptible to our human eyes. So we are not able to see the difference. What What Zarnik did, he developed such a device that adds the phase change lambda by 4 extra. So final phase change will be lambda plus lambda by 2. That is perceptible to our eyes. Okay. Here, two wavelengths. They are same wavelength, but their amplitude is different. A1 and A2, they are different. When the waves are in phase, that is A1 plus A2, the amplitude will increase. So the brightness of the light will be increased. And here, the light waves having different types of amplitude, they are having the same wavelength, but they are not in phase. So the destructive wave will be created. That will be A1 minus A2. A1 is having the greater amplitude, A2 is having the lesser amplitude, but A1 minus A2 will create much more lesser amplitude, so the dull image will be created. This is the optical elements of a phase contrast illumination system. Here phase plate is used with the objective lens, and phase annulus is used with the condenser. These two apparatus will create the phase change. Yeah. This is the diagram by which the phase contrast microscope, by the action of phase plate and phase annulus, creates phase differences. Okay. Here. This ray is passing through the gap of the phase annulus, and the phase change is 0. So the final change will be, phase will be x. That is its original wavelength. Here there is no change. Here it is passing through the gap, then passing through the specimen. Biological specimen changes the phase lambda by 4 extra. That is not perceptible to our eyes. So this is final change is x plus lambda by 4, because it is passing through the thinner portion of the phase plate. But here the light is passing through the gap and passing through the specimen. Here the phase change is lambda by 4, and then passing through the thicker portion of the phase plate. Here lambda by 4 phase change is occurred extra. So the final phase change is lambda by 4 plus lambda by 4. So the phase will be x plus lambda by 2, and this extra lambda by 2 is perceptible to our human eyes. So by using this microscope, we are able to see the movement of the microorganisms, and as well as we are able to see the unstained specimens also. The drawback of this microscope is it is producing basically 2D images, and it is costlier than conventional compound light microscope. These are some images. Now I am coming to the fluorescent microscopy. So many biological molecules are able to autofluoresce. That means they are able to emit fluorescent light. So with the help of this autofluorescence, we can be able to visualize them without any addition of fluorescent molecules. But those cells or those molecules that are not able to autofluoresce, we have to stain them with fluorescent molecules. And fluorescent molecule is having some property, that is, it can be excited with the help of lower wavelength of light, but this fluorescent molecule will emit higher wavelength of light. To see this type of phenomenon and to see the biological molecules that are able to autofluoresce or stained with fluorescent dyes, we have to use fluorescent microscope. Yeah. This is the fluorescence blue incident light. Okay. This is the FITC molecule. That means fluorescent isothiocyanate. Okay. And it is having the excitation maxima of 494 nanometer, but it is much more lesser, and it is in blue color. After excitation, it will emit the light that is having wavelength of 520 nanometer. That is green in color. This type of property is used in fluorescent fluorescent microscope. And this is the various types of fluorescent dyes that is having the excitation and emission maxima. DAPI 4,6-diamidino 2-phenyl di 2-diphenyl indole, then FITC fluorescent isothiocyanate, and TRITC tetramethyl B-rhodamine isothiocyanate. These are having different type of excitation and emission maxima. This is different curve of fluorescent molecules. Yeah, this is the basic pathway of fluorescent microscope. Okay, here this is the light source. Generally, tungsten bulb is used as source of illumination, and here it is excitation filter that filters the required wavelength of light. Here, the green light is able to pass, and other light is not passing, and it is then striking with the dichroic mirror. This is a special type of mirror. Okay. After that, it bends and passing through the objective lens and then to the specimen, where specimen is stained with the fluorescent dyes, or it may autofluoresce, whatever may be. After excitation, the light of having higher wavelength will passing through the objective. Some of the light is returning through the objective, then striking into the dichroic mirror, and then it is returning back. But the light that is having higher wavelength will pass through the objective, then passing through the dichroic mirror. After that, there is an emission filter here. Green light, then the light that is emitted is red in color, and this is passing through the emission filter, and that is going to the observer's eye. This is the basic working pathway of fluorescent microscope. These are the images of E. coli and other organisms of fluorescent microscopy. These are stained with different types of fluorescent dyes. This is fluorescent proteins, cell nuclei labeled with GFP, that is green fluorescent protein, YFP, yellow fluorescent protein, and RFP, red fluorescent protein. The drawback of fluorescent microscopy is it is costlier, and so many molecules are not autofluorescent. You have to stain with the fluorescent dyes, particular dyes. The image is 2D, and the image quality is not superb in comparison to its advanced version, that is confocal microscope. This is the banding pattern of the human chromosome and stained with the fluorescent dye GIMSA. So this is simply known as the G banding. And this is distinct patterns observed in human chromosomes with the help of banding pattern. Scientists are able to detect the chromosomal abnormalities in the human beings. And another applications of fluorescent microscopy are fish, then FRAP, fluorescent recovery after photobleaching, and various types of application, other applications are used. But the common applications are banding pattern, then observance of different types of cells, then freight, then FRAP, etc. Now I am coming to confocal microscopy. Okay. Yeah. Confocal microscopy was developed and patented by Marvin Minsky of Princeton University, USA in 1955 when he was a PhD student. And it uses optical imaging to create a virtual slice or plane many micrometers deep within the tissue. And it provides very high quality images with fine detail and more contrast than conventional microscopy. And the imaging technique allows for reconstruction of virtual three-dimensional images of the tissue when multiple sections are combined. So the various types of confocal microscopes are present. That is confocal laser scanning microscope, Raman microscope, tandem scanning microscope, and confocal theta microscope. Yeah, confocal, that means it is using the conjugate focal plane. Please try to understand. This colored light is having the focal point here, okay, and passing through this lens and this lens, and then there is a pinhole, and this light is able to pass through the pinhole. It is the focal point. It is its conjugate focal point. So it is using a conjugate focal point. So it is simply known as confocal. This is the basic principle of the confocal microscope. In a biological sample, that seems to be two-dimensional, but the microorganism is 3D in structure, and in a sample, thin sample, so many imaginary or virtual layers are present. Light may pass, suppose like that, This is the layers. Light may pass from here, pass from here, pass from here, here, as well as here. But their focal point, or sorry, their conjugal focal point is not at the same place. The light that is having the focal point in the pinhole only pass. Other lights, that's focal point, like in red light, that's focal point is not onto the pinhole, will not pass. So the confocal light or conjugal focal point light is able to create a better image, and the light that's focal point is not onto the pinhole will be discarded. So the noise formation is minimized, and the image is producing by the light coming from different layers of the specimen. By combining these thousands of images, a 3D image will be created. Yeah. This is the basic diagram of confocal imaging. This is the confocal microscope scanning unit. Yeah. To create an image of the specimen, the focal spot is rapidly and serially scanned in the XY plane, which is why the confocal microscopes yield horizontal virtual sectioned images. Okay. As the scanning progresses, signal from the detector is fed to a computer that collects all the point images of the sample and serially constructs the image 1 pixel at a time. Yeah. These are the components of confocal microscope: light source, that is laser, filters, acousto-optical device or IPCO disc, scanner, detector, that may be PMD or APD, then pinhole. These are the components of confocal microscope. Yeah. It is the confocal principle of AP fluorescence laser scanning microscope. Microscope or confocal microscope that is having dichroic mirror is simply known as AP fluorescence laser scanning microscopy. In simple term, confocal microscope is the advanced version of the fluorescent microscope. Yeah? The focal plane, the specimen, the light that is red in color is forming a focal point at the pinhole is only passing. The light that is coming from different point, that is in green in color, is forming focal point in another place apart from the pinhole aperture. So this type of light or this type of image that is formed, a noise is discarded. Only the light that forms a confocal image on its pinhole will pass and create image. This is the basic diagram. Yeah. Please try to understand. Yeah, আছে নাকি? Laser torch আছে নাকি? Hello? Laser torch আছে? Please try to understand. Laser It is the source of illumination. Light is passing through the emission filter, then it is striking onto the dichroic mirror, and these are the rotating mirrors, that forms the different images. Okay, and after that, through the objective lens, it will come to the fluorescent sample. After that, it will excite the fluorescent molecule and emits the higher wavelength of light. And this is the green in color. Then it is returning back through the same direction. Then after striking to the dichroic mirror, it will passing through the screen with pinhole. Other images where its conjugate focal plane is not onto the pinhole will be discarded. Only the light that is having the conjugate focal plane onto the pinhole will pass and form the image that is detected by PMT. This is the basic working of the confocal microscope. And this is the argon laser that is producing different types of light having different wavelength. And this is the scan control mechanism in a confocal laser scanning microscope. Yeah. Please try to understand. Laser light is passing through the microlens array, and then there are so many holes onto the microlens array. Then it is coming to the dichroic mirror. Then it is passing through the pinhole. Okay. That is simply known as the NIPCO disc, and it is the 50 micron pinhole array. After that, it is passing through the objective lens and striking onto the fluorescent molecule present on the specimen. After that, light that is emitted through the objective lens, then through the pinhole. Okay. And it is spinning. So it can scan thousands of images. By using that pinhole, only the light that is having the conjugate focal plane onto the pinhole will pass. Light may be coming from different depths of the specimen. So their conjugate focal plane will be at different space. Only the light that is having the conjugate focal plane onto the pinhole, or that is NIPCO disc, will be able to pass, and it is spinning. Okay. And by this way, thousands of images are created, and then it will strike onto the dichroic mirror, then barrier filter. After that, it will be catched on the monochrome CCD camera. Thousands of images. And this is the same diagram of simpler version. Fluorophores. The choice of fluorophores is the most important aspects of fluorescence confocal microscope. It is typically influenced by several factors. The fluorophores should tag the correct part of the specimen. It must be sensitive enough for the given excitation wavelength. For living specimens, it should not significantly alter the dynamics of the organism. And fourth is that the specimen should not affect the fluorophore, as its chemical environment can affect the position of the peaks of the excitation and emission spectra. Yeah. Transverse XZ or YZ cross-sectional views can be generated by most confocal software programs. The object appears as if it had been cut transversely, that is in a plane oriented parallel to the optical axis. Now, 5-dimensional views including information in X, Y, and Z dimensions in a timed sequence and in multiple colors. Okay, this is the confocal diagram of triple stained rodent embryo tissue. That is, here it is the mouse embryo and it is rat embryo. Yeah. The quality of the confocal images can be determined by four principal factors: spatial resolution, resolution of light intensity, that is dynamic range, S by N ratio, temporal resolution, etc. Yeah. Please try to understand. It is the image that is taken with conventional fluorescent microscope. The image is much more inferior quality. And here, the image is taken with the help of confocal microscope. Same specimen, but the quality of the image can be seen with your eyes. You can clearly understand. Yeah, that is the Euclidean space. Suppose, in case of confocal microscope, you are talking about the 3D imaging. I am giving an example. A book on cell biology or microbiology, whatever may be, is having the same length and same width, but one book is having 300 pages, another book is of 1000 pages. Which book will contain more information? You will easily say that, sir, the book that is having 1000 pages will contain more information. Please try to imagine one page is one scan. 300-page books is having 300 scans. The book that is having 1000 pages is having 1000 scans. That contains more information. Euclidean space is the imaginary space that was proposed by ancient Greek mathematician Euclid of Alexandria. That is the axis, x-axis, y-axis, and z-axis, a typical 3D diagram or 3D box. Okay. The voxel concept in confocal images. You are aware about the concept of pixels. Pixels are picture elements that is present in X and Y direction. But the voxel, that is volume of pixels, that is having a 3D conformation. Okay, that means X, Y axis along with the Z axis. Voxels are the basic spatial unit in 3D confocal imaging. I am just trying to explain you. Yeah. It is simply a diagram of a box. The x and y direction. Suppose that these are the pixels. Each layer, if it is one layer, it is one layer having pixels. This is another layer, this is another layer, another layer, another layer, another layer. If the layers are having same quantity of pixels, or the layers having different quantity of pixels, so the voxels can be divided into two types of voxels: isotropic voxels, voxels having equal dimensions along each axis are called isotropic, whereas having different dimensions, at least one of the planes are called anisotropic voxels. Okay, and each voxel in an image has a unique digital address within 3D space, X, Y, and Z. Okay, so it is pixel XY direction, it is the Z axis. So different layers is having same type of pixels is isotropic voxel. Different layers is having different numbers of pixels are simply known as anisotropic voxels. Yeah, please try to understand. This is X and Y, simply known as pixel, and it is Z. So the X and Y axis here, so many imaginary layers may be present. If the XY pixels in each layer is same, then the voxel will be isotropic. If the pixels in the different layers, at least one layer is different from other layers, it will be anisotropic. And this is typical raster image that is used to explain the pixel. Okay, and this is the 3D imaging of eukaryotic nucleus, where the focal depth, that is voxel, 500 to 1000 nanometer. Here it is bacterium, 1 to 2 micron, virus near about 200 nanometer. Z stacking. To create an image of the specimen, the focal spot is rapidly and serially scanned in the XY plane, which is why confocal microscopes yield horizontal virtual sectioned images. Here the sectioning is done virtually, not like the sectioning that is done in transmission electron microscope with the help of ultramicrotome. Here only the virtual sectioning of the specimen is done, and thousands of images are used to create a 3D image. And the stacking, that means from bottom to top, the layers are stacked like the pages of books in the direction of a Z-axis. So it is simply known as Z-stacking. Yeah. Please try to understand. Frame in a Z plane. Here, this is a layer, this is a layer, this is a layer, this is also a layer, this is also a layer. And these are the XY plane. So, by uniting these thousands of layers, a 3D image is formed. And this is in the Z direction, so it is simply known as the Z stacking. Yeah, please try to understand. This is lambda stack, XY direction, and this is the block, how X, Y, and lambda is shown here. This is the sectioned images in 3D, that is Z stacked image. Different images are taken with the help of confocal microscopes, and the number of images may be of thousands, and these images are united with the help of software that is used for Z stacking, and that will create finally the 3D image. This is another diagram how the 3D imaging of confocal microscope through Z stacking is used to create a 3D image. Yeah. Advantages of confocal microscopy: high resolution, high contrast images, reconstruction of 3D images, absence of artifacts induced by conventional microscopy, in vivo microscopy to a skin depth of about 200 micron, etc. Limitations: high cost relative to conventional microscopy or dermoscopy, photo bleaching of fluorescent probes and phototoxicity of live samples, etc. And depth of imaging is limited by optical penetration and signal to noise ratio. In vivo confocal microscopes can create or generate high resolution images of the entire epidermis and a superficial layer of dermis. These are the applications of confocal microscopes, that is FRAP, fluorescent recovery after photo bleaching, FRET, flame, flip, flap, fish, and live cell imaging. I am not going to the detail of the techniques, that requires a different lecture. With this, I am ending here. I think you have received an idea about the light microscopy. You are welcome to the world of microbes with the help of microscopes, and you can explore it, you can read it. So many reference books are available. I am just telling 2-3 books. Books on light microscopy by Douglas B. Murphy. This is an excellent book. Book by Alan J. Lassie on light microscopy. That is also excellent book. Book by light microscopy and cell biology by V.K. Sharma is also a good book. You can go through these books, explore it. You are welcome to the Department of Microbiology, Barakpur Ashtaguru Surinath College. You are also welcome to the world of microbes. Thank you very much.